think I'm going to go ahead and get us started. Welcome, everyone. This session is being recorded. You are at the webinar on Translating the Complex Genetics of ASD with Dr. Ed Cook from the University of Illinois at Chicago. And this is presented by the Autism Program of Illinois. Welcome. My name is Mary Kelly. I'm the Quality Coordinator at the Autism Program's Central Office located in Springfield. Next slide. So this is our mission statement. I'm not going to read it to you, but I want you to all know that the Autism Program of Illinois is a state-funded grant through the Illinois Department of Human Services with 15 network partners who throughout the state support children and families who have an impact with autism, from early assessment to treatments and other services and support. Next slide, please. This is a map of our service locations. As you can see, we span the state from Rockford down to Carbondale. Many of our TAP network partners also operate multiple site locations. So you may find that they have several different locations in the state. And this way we attempt to serve everyone throughout the state of Illinois with autism services. Next slide, please. So there are several types of services that we provide. Like I mentioned, everything from autism screenings to diagnostic assessments, therapies, group programs, family and community resource supports, family services, and education and training. Many of our partners do their own training programs, and we, the TAP Central Office, also conduct trainings such as this one brought to you today. We are going to have an opportunity for questions and answers, so please put your questions in the Q&A box. There will be a survey that will pop up at the end for you to complete, and I will also put that survey in the chat link as well. The survey is very important so that we can continue to provide these great trainings to individuals and community members in the state. You will want to indicate on that survey that this was provided by TAP Central Office and that it is a training. Next slide, please. It's really hard to see with all that purple stuff. Oops, sorry. There we go. Just wanted to let you know that we are on the web at tap-illinois.org. We hope to be launching our brand new website next week, so please check that out. It's really fabulous. We're also on Facebook, Twitter, and YouTube, so you can find us there as well. So again, put your questions in the question box. Dr. Cook will be monitoring that throughout and answering questions as he goes, and I will assist him with that. And then I'll have the survey link for you in the chat box as well. So without further ado, I am going to turn it over to Dr. Cook, and he will introduce himself and get us started. Thank you.
Thank you very much. Let's see. No commercial disclosures to report, and we missed the title slide, and just say a couple things about myself. I'm at the University of Illinois at Chicago, child psychiatrist, also been engaged in much of the research on autism that I'm going to present to you, where autism has moved from knowing that there are genetic contributions to now having some findings that are of clinical relevance. So we'll start with, why did we ever think that autism and genetics might be connected? And some of these numbers have changed over time, but this is the current status. The risk to siblings, this would be full siblings. This would be the answer to a couple that had a child with ASD, wanted to know what the risk would be that there would be another child with ASD, and the answer is 1 in 5. Given the population prevalence is about 1%, that's a 20-fold increase. We also know that if 2 are affected in a SIBSHIP, or 2 children, then the next children will have closer to a 1/3 to 1/2 recurrence risk. That's the first indication of what will be a theme in this presentation, that the genetics of autism is complex. That was in the title. And this is one example that the risk to all families is not the same. Now, things can run in families but not be genetic. The language that we speak is very familial but not at all genetic. So then twin studies are extremely helpful to separate this out. And with the current definition of autism spectrum disorder, if one identical twin has autism, then there's a 90% chance that the other identical twin will have autism spectrum disorder. I don't know if I said, if the first one has autism spectrum disorder, there's a 90% chance the identical twin will have autism spectrum disorder. I'll probably start saying ASD. There's no or a much smaller increased risk to non-identical twins and siblings, and genetic contribution estimates range from 50 to 90%, but the overall analysis, if you put all the studies together, is closer to a 90% genetic contribution. So genetic etiology groups, I'm going to probably pause after this one because there's a lot sort of embedded on this slide. I try to clarify some of this with the size of the type. So there are 2 kinds of ways that genetic acts, and one is de novo. It's not inherited from either parent. These are rare. They're predicted in genetically lethal disorders. What that means, genetically lethal, because I want to try to always say what that means, is it a disorder that affects the chance of having children? So clearly, some people with ASD have children, but there's a reduced chance of having children if you look at everyone with ASD, and this for disorders that don't disappear, there's usually this spontaneous rate, you could think of it, where autism basically arises in either the sperm or the egg and wasn't present in either parent. Now, these are where we find the largest individual effect, and if we think about clinical relevance, it's these rare de novo or out of the blue mutations that often have a large individual effect, or we might say is what's contributing mostly to why someone has autism spectrum disorder. If you add all these up together, and there's some differences in numbers, my 10% number is probably conservative. I'll talk more about that later. Some people think it may be a higher impact, but certainly these ones that are rare, large effects that we're more or less sure about is more in the 10% range. Now, the inherited, I would say that we kept looking initially for sort of common inherited variants, but if we look back at it, the common variants, I'm going to skip down a little bit to common variants, they're generally a very small individual effect. And so the most recent paper was in 2019, mostly because of the technology we were using. This is what we were looking for initially. And the point is that if you add up a lot of these small changes, we all have many places of variation. Most of us are very similar to each other as humans. But if you aggregate all these very small individual effect variants, this probably accounts for about half of the overall autism risk. And this we'll get into a little bit more. You can have rare inherited effects, and these are typically from recessive and recessive where both parents are unaffected, so they can easily transmit the risk alleles, but the child gets the risk allele from both parents and now has it on both chromosomes. So those can be a large effect. I'll talk later about how much of that contributes to autism. It's more in the range of, it's actually more in the range of about 1 to 2% of all ASD. I'm going to stop there for questions. I don't want to lose people right at the beginning. I don't see any in the Q&A box. Okay, we'll move on. Oh, I got a question. Thank you. If a couple has only 2 children, but they are identical twins, and both twins have ASD, what are the odds of a third child having ASD? Would it be 20% or higher? That's an excellent question. And we consider having twins that are identical as basically counting as one in the sense of genetics. So in this case, that the chance of the next child having ASD would be more as if there had just been one child born with ASD. It's a really good question. Okay, so this is a genetic model of autism. And what I want to show you here is this over here would be most cases of autism. And we would call them multifactorial, which means lots of genetic variants and probably lots of environmental effects. And I'm going to pause a little bit here and talk about that. This model here is genetic etiology, and it tends, you don't talk about inherited environmental effects, although you could think of them in some way. But the point I want to make about this idea that it's the rare events that are big effect in genetics, and you otherwise have mostly these small little effects. And by the way, those small effects are both risk and lack of protection adding up. You could think of a long equation of adds and subtracts toward risk. And the same thing probably happens in environment. Everybody's always looking, as we were doing, for common genetic effects that would contribute to a large part of autism. That doesn't happen. That's probably also the case in the environmental component of autism. People want to look for something that's a big effect across all autism. Those tend not to be found. And so now in the environmental work, people are starting to look at combined effects of various environmental effects and not just one big one, although there are some one big one environmental effects, such as exposure to valproate and anticonvulsant during pregnancy and very low birth weight are analogous to the at least infrequent large effect environmental effects that we know about. I'm going to answer this question because I think it's related. Working with identical twins, one has autism, one does not. Yeah, that's not always the case, but I'd also ask, the question is, are they both completely discordant? And the other question is the identical status confirmed with genetics. But yes, not every case. I mentioned 90%, so certainly not 100%. My nephew has Asperger's. His sister has Downs. Both live with me. Their parents have both passed away. We did family counseling. After my husband passed away, I spoke to the counselor, and she felt my husband also symptoms for Asperger's. Is there any connection? Certainly there's not a connection probably between Down syndrome and Asperger. and you know, it's a very good question about Down syndrome, because Down syndrome is almost always de novo. And if we go back to the de novo piece, I know there are two parts to the question, but let me talk about, I thought about mentioning that earlier. So Down syndrome is really the first rare de novo that we knew about. So usually 3 chromosome 21s, and comes from parents with only 2 chromosome 21s. That tends to be de novo. There are some rare cases of Down syndrome that are due to a translocation that can be inherited, but for the question, we're going to assume it's typical genetic cause of Down syndrome. And that's not going to be something that is related to Asperger. Although if the sister that has Down syndrome is at somewhat higher risk of ASD. It's a genetic syndrome that's not highly related to risk for ASD, but some risk. But the connection, if you're thinking about genetic counseling, they would likely not be connected. The nephew having Asperger, and that's the nephew, and then so what I don't know, maybe you can put in the question, is whether the nephew is your husband's son or your son, or your I don't know if that makes any sense. Wait, it wouldn't be your husband, be your husband's sibling's son, or whether it's your sibling's son. I probably confused the question. And I'll just make a comment on this one. Either way, is that this is very important, that the risk for, because of what I say down here, assuming now here's what we're getting this very complicated question. This is exactly why it's a question for a genetic counselor and not the talk. I'm sorry to say that, but I want to say a couple of general things about it, which is that your husband's sister's son. Okay. So there may be a connection. The easiest way to know if there's something relevant for you is actually the testing of the nephew. But yeah, again, full counseling. This is most likely going to be related to this, what we're talking about, these common variants. And there's no real clinically applicable way to say that there may be something related. Well, husband, sister, yeah, there may be something related between the two. And it could be related to husband, sister, son. I'm just trying to figure this out. Yeah, it's a complicated one. It's worth more counseling. And so let me talk about what typically happens versus the specifics that would be best done with counseling and testing in the family. Is that what's often happening is there's a polygenic risk, or these many variants, and the variants also interact. As we know, there are 4 times as many males than females with ASD. There's something we think is protective about being female, which may relate to the to your husband's sister not being affected. This could possibly be, and just without seeing the pedigree in front of me, could possibly be something X-linked. And because she's not affected, but her brother and son are, that's something to look for. Typically, Fragile X is probably more severe than describing as Asperger, but that would be what we currently would be testing for of the things that might be X-linked. At the very end, I'll talk about what today we would do in terms of testing. Okay, I'm going to move on. Thank you. So this is a genetic model of autism. So I was talking about most cases being multifactorial. We used to think it might just be 5 of these relatively common variants that would contribute to autism if they were all present. Now you could think of these circles as hundreds of variants all sort of converging to contribute to risk. And I think we try to show here that the range of severity might be related to the proportion of variants that someone had. So this is most cases, and actually a circle might be a set of environmental risk adding up. And then there's a whole nother set that I tried to make smaller. This is 10 to 20%. Some people say it may be higher. I'd probably be on the 10% range. And these are less complex. You notice I don't say simple genetics. These are almost all of this rare de novo mutation type. So examples are maternal chromosome 15q11, q13, having an extra or duplicated or 2 extra or triplicated copies. This is about 0.5 to 1% of ASD. Chromosome 16, the small arm, very close to the center of the small arm, either deletion or duplication adds up to about 0.5 to 1%. Fragile X I mentioned before, about 0.5 to 1%. And then I'm going to, one of the first ones we knew for sure was a single gene loss of function SNV, and I'm going to explain what an SNV is. And now there are many other, these are actually called CNVs, and I'll explain what those are, and now there are many others. But the point is, most are still in this multifactorial range, but some are in this less complex range, and I'll go into more detail. So chromosomal disorders, insertions and deletions. So these are not like Down syndrome. You have a whole extra piece of a chromosome, of chromosome 21. The only other full change in a chromosome is a absence of the X in Turner syndrome. So most of the changes in chromosomes from the early days where you could see it under the microscope was a piece of a chromosome, either missing or an additional copy. Here are some of the most common deletion syndromes, and they typically range 1 to 5,000 or less in the population. Williams syndrome, 7q11, 15q11, q13 deletions, Prader-Willi syndrome and Angelman syndrome, and duplications, which I'll talk to you about maternal duplications, which are a risk for ASD. 16p11.2, 22q11 DeGeorge or VCFS, and 22q13, which leads to Phelan-McDermid syndrome, which is one of the, through syndrome, seeing changes in features was one of the, and also basically from finding the deletion. It is an autism syndrome. So of some of these most common chromosome changes, 7q11 duplication, not deletion. The deletion leads to Williams syndrome, which at least early in development has increased eye contact, increased sociability. In autism, it's the opposite, or the duplication of that region. It's not always a mirroring like that, because in 16p11.2, either the deletion or the duplication can lead to ASD. I've mentioned, and we'll mention more, this 15q11, q13 duplication, and 22q11 deletion duplication has ASD risk, and this 22q13 Phelan-McDermid syndrome is an ASD-related syndrome. Okay, what I'm showing on here under Roman numeral 1 are 2 normal chromosome 15s, and I'm putting an arrow. I could put the arrow in the same place. This is the chromosome that comes from the mother. This is the chromosome that comes from the father. This is a region of Q11, Q13 that if it's missing from the mother, it causes Angelman syndrome. In other words, the chromosome that came from the mother. If it's missing from the father, exact same missing piece, it leads to Prader-Willi syndrome. And Prader-Willi syndrome is mild or intellectual disability or not intellectual disability in terms of more severe learning disability, very consistent severe obesity and OCD in addition to any any uh, uh, any overeating, for example. In Angelman syndrome, deletions, it's missing from the mother, and severe or profound cognitive impairment, intellectual disability, and consistent ASD symptoms as well. This is a patient that I've seen for a long time, and this is an extra piece of a chromosome. So let me go back and show you that again. So this patient I'm going to show you has an extra piece. Oh, I didn't show you the duplication. I showed you deletion. The duplications can be interstitial. So this is an extra piece of that region on the mother's chromosome, or it can be an extra chromosome, so 47 chromosomes. As I show here, these tend to have two extra pieces of this region. So now this is the patient that first got me interested in this, and I direct you to this duplication 15q alliance. One of the best ways to know that it's an ASD related syndrome is that most of the questions that come up there sound like they'd be questions for a typical ASD group. This is a woman with autism, severe intellectual disability. This syndrome in particular frequently has seizures. This group of patients with this extra piece of a chromosome coming from the mother often have repetitive behaviors, anxiety about lack of order, and mood lability for quite a few. This woman had self-injury, and she's had a good response to an anticonvulsant, both for mood stability and preventing seizures, and to an SSRI medication to help treat the anxiety about lack of order that seems a lot like OCD. Again, this is 0.3 to 1% of ASD, and this is how we found this sort of extra piece. And this is what you should only have 2 chromosome 15s, but she had this extra piece. This is looking at a duplication, and this is, you should only have 2 pieces of this. Actually, this is just another marker. It's not the region. You should only have this region near the centromere. You see 2. In this case, you see one of them here because it's near the teal blob. And then over here, you should just see one thing near the teal blob, but you see 2. And that is an interstitial duplication of that region, but still 46 chromosomes instead of 47 in the one that we saw before. So this is the pedigree showing, actually what I showed you with the fish could have either been the 2 children with ASD, or it could have been their mother. And so I won't go into exactly how this numbers tell us where the chromosome came from, but maybe I'll try to walk you through that. This is the mother's mother, and she has markers across this region. You see she basically has 2 chromosomes. So she should pass one of this set of markers on to her daughter. And she does. She passes this 197, 131, 98 to her daughter, the mother of the children that she has with ASD. And the father should only, therefore, bring this in over here or bring that in. But in the development of the sperm, Both of these come down. So instead of 197, 139 or 197, 147, you get 197, 139 and 197, 147 because you've duplicated that region and brought both of that region to here. So now you have 3 copies of this region, which is typical of the interstitial duplications. And now the mother gives, now it could go either way. She could give this that came from her father. I mean, she could give this that came from her mother, or she could give this that came from her father. Now, she gives these 2 children the extra piece, so they both have the interstitial duplication coming from their mother. Now you go, why isn't she affected? Well, hers came from her father, so she has lower risk, almost no risk. The children therefore have a risk. This is an exception where it's not de novo from the mother to the children. It is an exception. Usually that's the way things work. In this case, it's what's called a parent of origin effect, similar to Prader-Willi and Angelman being different is why she's not affected, but her children are. She actually has had 2 daughters now, both of whom do not have the duplication and are not affected with ASD. She actually did have, this was found after these 3 children had been born, but based on this, she was able to know whether she made a decision about having a child later, and she would not have gotten pregnant with the 4th child if she didn't know that she would be able to test and be able to make a choice. She didn't, I'm not sure what choice she would have made, but the other child did not have the duplication. This is that region. So one of the things when we find these extra or missing pieces is that we look at the region and see which genes are involved. So there are several genes in this Prader-Willi or Angelman region. For Angelman syndrome, it looks like most, but not all of Angelman syndrome is related to this gene, the UBE3A gene. And probably most of these kinds of syndromes where you have, this is about 4 million base pairs of DNA. Most of them, there are more than one gene contributing. And in this region and in most of these kinds of findings in autism, we're not sure exactly which gene it's pointing to. And I want to step back and say these chromosomal insertions and deletions are also referred to as CNVs or copy number variants. And I'm going to define that now. So a copy number variant is an insertion or deletion. It's like a missing or extra paragraph or page of a book. So it's like you could think of the chromosome as a book, and this deleted or extra piece is an extra page or missing page. These are not always contributing to some kind of clinical problem. Obviously, these have risk for things other than autism. They're typically, especially related to autism, relevant if in that missing page there's a gene, because in that chromosome there are many pages that don't have genes, don't make proteins at a relatively silent areas. I did say relatively silent areas. And I've used this term de novo a lot, and hopefully it's clear that it means it's new. It wasn't present in the parents. So I'm going to stop, see if there are questions. We were actually looking for common variants when we found a bunch of CNVs in autism, and that all happened around the mid-90s and consolidated with clinical testing early in the last decade. Okay, so autosomal dominant, people probably can think of Huntington as the autosomal dominant that most people probably know about, where half of the children have Huntington, if the parent did, it gets passed on in that way. And therefore, you'll see it in many generations. You'll see half of the offspring having it. You don't see that in autism pedigrees because there's often not offspring to have the long multi-generational. If you think about why you see it in Huntington, Huntington is very severe, but it's not having an effect on being passed on because it's passed on before the severe illness develops later in life. So again, de novo autosomal dominant, not present in either parent, occurs when the severity of the disorder is enough for less likelihood of having children, and it accounts for a child to have a severe clinical condition with unaffected parents because neither parent has the mutation. And this is what the pedigree looks like. It's not a big, large pedigree. It's sort of a paradox because it's not this large pedigree where half are affected. And what the dominant refers to is it only takes the mutation on one of the two copies, as opposed to one of the two chromosomes, as opposed to recessive, where it would have to be present on both. And recessive isn't going to happen de novo because it would be, these are rare events. And to put it in context, all of us on average have one rare mutation that isn't present in our parents, but is present in the DNA that codes for our proteins. The difference between whether that change, that sort of random change, has an effect or doesn't have an effect is where it occurs in the gene, which genes it occurs in. And the point is there's not an increased rate of mutation leading to autism where it's present from this mechanism. It's just in some sense, we all have on average one of these, and it's just a question of, in a sense, where does the lightning strike? Does it strike in a place that matters in developing the brain, or does it strike in a place that, you know, maybe there's, and I'll show you this later, there's some genes that you don't need two copies for functioning. This is the 16p11.2 I talked to you about, and shows you just a little bit graphically that you're looking across chromosome 16, and this is a 1-to-1 ratio between a control sample and this, it was actually an agree subject from one of the first genetic collections where there was a deletion of that region. You see there's only in that region closer to half as many, so there's only one copy of that particular region. So it shows you this missing piece across chromosome 16. You may wonder why there are no dots here. That's just around the centromere where they just sort of say there's a bunch of million bases there. And this is zooming in, very similar to what I did with the duplication on 15. And this is from the paper where our group showed this deletion. And what we were very interested in, again, first of all, this shows you where it is on chromosome 16. And also, so you see all these genes, and the question was, in this region, which are the genes that are causing ASD? In that particular paper, we found, I think, about 3 or 4, I think it was 4, with 16p11.2 microdeletion. So then we would take hundreds of samples and sequence them to see if we would find something in cases with ASD versus controls. And the answer, like many other of these that I mentioned to you, in contrast with UB3A and Angelman syndrome, is that we couldn't find any of these specific genes contributing. So it's probably a set of these genes missing that's contributing to risk. Now, when we first found this had only been found in autism. And it's easy to jump to the conclusion, okay, well, if you find this, then you have autism. But if you only look for it in autism, you're not going to find it in, well, we didn't find it in controls. but that doesn't mean you're not going to find it in people with something else. So, and this has been extremely well studied by the Simons Foundation, 100 people with this deletion and 100 people with duplication. And this leads us to variable expression. So with these deletions, you can see ASD, ASD with intellectual disability. You're always going to see obesity with the deletions. There's no variation in that part of the phenotype. You might have ASD, ASD with ID. Actually, with the deletions, you can also have language disorder without ASD or ID. The duplications, ASD or schizophrenia. Now notice not schizophrenia with deletions, and some people wanted to say, well, you get deletions, ASD, and schizophrenia duplications, but okay, that's fine, but you also get ASD with duplications. Exactly why we don't know. Notice I underline thinness again, because the one thing that is consistent is that as your numbers of this region go up, your thinness increases. So it's actually quite severe obesity. One of the reasons to know about 16p11.2 CNVs, for example, we found one of the research was done in a way that we could give clinical results back. And what was important in the counseling is saying, well, all the work you've been doing in adolescence and before to keep from obesity will be even more important going into the 20s, because it's probably brain related, that the brain's basically saying signals to be at to be at homeostasis, or to be at the point the brain thinks the body should be at a higher weight. And then the opposite is thinness, with some that are so thin to, at least based on their body weight, to be in the category of as thin as having anorexia. Both of these are rarely seen in controls, so their variable expression is one term that you can see different manifestations in different people. Penetrance means if you see it, do you then, if you have the deletion, do you have a problem in this larger group? This one is highly penetrant. People with 16p11.2 deletions are affected by it, but affected in different ways. And this becomes very important because let's say you found this you found this right after birth, for example, you wouldn't be able to predict those outcomes. You wouldn't know who's going to have ASD or severe language disorder without ASD. You might be able to predict in terms of risk for obesity or thinness, but you know something's going to happen, but you don't know the level of severity. As it turns out, even though we found it in autism, it's only at about 25% will have autism with this deletion. What's interesting is the Simons Foundation studied the parents, and the first thing that was notable is the range of IQ was basically shifted 2 standard deviations lower. So in some sense, a 30-point IQ drop. So instead of 100 plus or minus 15, it was moved to 70 plus or minus 15. And what they found out is the ones that didn't have, that basically had 100 IQ, had parents with an average IQ of 130, obviously not so common. And the ones that were lower than the average of 70, at the lower end, had parents that normal IQs, but the difference would be 30 points lower than 85 versus 30 points lower than 100, as an example. That partly also tells you that the multifactorial impact of, and this is a good example of, even though I gave you this slide that showed this sort of polygenic component and this separate chromosomal changes, that the two actually interact. So this is actually showing an interaction of the multifactorial piece related to IQ with the effect of the copy number variant. This, I'm going to try to explain this one, but it basically shows that some of these CNVs have a stronger impact that by themselves are probably enough to be enough risk to give somebody ASD. None of them are 100%. If you have them, you have ASD, and that's because there are other genetic factors and non-genetic factors that can either make it more severe or less severe. And so in the case of the 16p11 example that I gave you with the deletion, it also related not just to cognition, but also to how far the parents were on. We are all on the autism spectrum. Some of us, I'm not going to say me because I'm on the autism side of the autism spectrum, but there are some on the other end from what we diagnose as autism spectrum, and the people on that other end with better than average social communication function and more flexibility, actually, they're the ones where the risk for ASD actually is reduced if there's a 16p11.2 deletion. It's a very elegant paper I can send to anyone that's interested. And then we have things like the paternal duplication of that 15q11, q13 region, which if it's leading to ASD, it's probably other factors, not by itself. And then you have ones that are kind of intermediate. I will point out that there are deletions that only include SHANK3, not this larger 22q13, and there are deletions that only include orexin-1. And they've been some of the early ways that people have been studying the biology of autism because you had it honed down to a single gene. So I talked to you about CNVs, or copy number variants, and now I'm going to focus a lot on SNV, or single nucleotide variants, which have even more changed our understanding of how genetics leads to autism, and I think is the most promising avenue to where we're going to learn more about autism and hopefully develop some improved treatments. So there are two kinds of single nucleotide variants. One is missense. It changes an amino acid of a protein. So all our proteins have one of a set of amino acids along a chain, and that's how they're defined. Loss of function, or LOF, I'll be using a lot. The change in the nucleotide changes an amino acid to a stop codon, or it affects splicing that leads to a frame shift of coding. So for both types of loss of function mutations, an example would be a mutation that instead of coding for the 19th amino acid, codes for stop making this protein. And so instead of, let's say, 400 amino acid protein, you have an 18 amino acid protein. And typically those just don't function as well. So loss of function will often imply that instead of two copies producing the gene, you only have one copy, and is basically reducing the abundance of that protein in half. So here's an example. With de novo SNV, both parents have supercalifragilisticexpialidocious. With a de novo missense, in one of the child's two copies, again, you can think of each letter as an amino acid and the word as a protein. They're going to have supercalixragilisticexpialidocious. Clearly not the same thing, but most of it's there. And the question becomes, is the substitution of the F for the X? You can kind of see in speaking it, it was kind of hard to say. There are other changes you might make that might not make it as hard to say. And that's where some of these missense mutations impact or are deleterious to that protein, and some are not. Now, in contrast, the de novo nonsense, or loss of function, the analogy is instead of an X replacing the F, there is a signal to say stop making this protein, And so you just get supercali. And supercali is almost certainly not going to function in the way that this larger one does. Questions here, because what an SNV is, I think, is important for what I'm going to say later. And we haven't had questions for a while, so I want to see if someone's still awake. Everybody's looking for their Q&A button. Okay, I'm going to move on and I'll sort of monitor. So in the same way that we didn't find CNVs until the technology developed, and most of the technology for this, really, you couldn't do this without the Human Genome Project finishing around 2000. So that gave us, when I show you the chromosomes and the base pairs, we really needed the Genome Project to be completed, and there's been technology since then. The most recent has been whole exome sequencing. We're about 10 years into that. And to tell you the truth, we started whole exome sequencing looking for variation that would be similar across many people with autism. And once again, we found de novo variation being what was driving things largely. So I now say the emphasis has been finding de novo SNVs in trios of individuals with ASD and their parents. So the best way to know if there's a new mutation is to both sequence the parents and the child and see what's different. And I already said that that's going to be different about once per trio of parents and child. So each child on average within their protein coding region, which is a subset of the genome, is going to have a change. And many of those changes will be silent. Some will be loss of function. Some will be missense. So what was found is that there was a higher burden for loss of function de novo SNVs and a smaller enrichment of de novo missense SNVs. So the big effect here, the big signal is in loss of function. There's some difference in just changing the amino acid in the protein. And this is key. It kind of makes sense that it's only going to contribute to ASD if they're in genes expressed in the brain. I mean, I suppose there are other possibilities for autism can happen, but this is consistent with the idea that something is happening in the brain in terms of contributing to ASD risk. The other thing is the genes have to be intolerant of missense mutation. So some genes can tolerate missense mutation. Actually, some genes can tolerate loss of function mutation. So there's a higher burden in not just for loss of function, but also loss of function in brain expressed intolerant of loss of function. But you only really see the signal because it's weaker in the de novo missense that are in brain expressed genes that don't tolerate mutation. So some genes need to be much more exactly as they were and can't tolerate a change in amino acid. And there are quite a few changes that can be made. There's actually a browser that can be used to determine which genes are tolerant for loss of function or missense mutation. And I would comment that the Broad Institute has been very important in the last decade of this work. They've done most of the sequencing and maybe change, well, no. Yeah, they've done most of this work and just developed the capacity to do it. And as important as sequencing, the tools to analyze the sequence data. This is the most recent paper showing the SNVs that are significant for knowing statistically that they're contributing to ASD. This was published about a year ago, almost exactly. And many of these genes at the top we've known about for a while. Now, just to give you a sense of how significant this is, this Q value roughly like a p-value. It's not exactly the same. This is like 10 to the minus 16th significance value. This is the 0.05 level. So everything above here you would consider conservatively of what we use in large experiment or any experiment of 0.05. So it's a bunch, it's not one. So that's the first thing you're struck with here, right? So if we were looking to 2 to 5 genes, we're not finding them. By the way, this is spread from the chromosome 1 through chromosome 22. There are analyses of X going on. They're harder to do. Yeah, it's a lot of genes. And 102 genes that we talk about includes this group below as well. So let's talk about, if you add up all the things I've been talking about, what is the rare burden? These are rare events contributing to ASD, and these are, this is the numbers you come up with. So exonic de novo CNVs, so that's what I said, if they include exons or part of a gene, about 5% of ASD will find those. Those, as it turns out, just as a group, tend to have the highest risk relative to unaffected siblings of about an odds ratio of 5 to 1, meaning you'll see about 1% of unaffected siblings and 5% with ASD. So quite an increase in risk. Loss of function de novo, SNVs, 10%. That's a large number, but there are also more controls that have those. And then what I mentioned before is these, they're rare, but they're inherited. The autosomal complete loss of function, which are best thought of as homozygous. These are about 3%. And then rare X-linked homozygous, meaning that if they're on the part that's specific to X, if males have something, they do not have another X. So they become the only copy, so about 1.7%. So you can say, well, add that up, you get about 20%, but the problem is if you add all that up in controls, you get about 10%. So the net increase is 10%, but you have to individually sort out the relevance of each variant. It's easier for the ones that recur rather than the ones that might occur only in one individual. Now, there are some large CNVs that are large enough that clinical geneticists will decide, even though they're the first patient being seen, that they are relevant. But it's hard when you only have one case. You can't make a syndrome with one case, but some of these are of such of impact that even though that gene or that region hasn't been deleted, or particularly regions impacted, it can be sorted out. It's a complex process. There's no simple answer to that question. So, I don't know, a long time ago, like 95, I gave a similar talk and said, we're going to find one gene and we're going to study that gene really well and we're going to develop a treatment for it. And it's obvious looking at this table that it's not that simple. We have many genes involved. So this becomes a question. There are many individually rare CNV, SNVs. Few of them are recurrent. Will we need different treatments for each CNV or SNV? And so what a lot of people are focused on since then, and frankly, the science of autism genetics right now has a lot to do with the people that know how to manage vast amounts of data and basically figure out, is there a commonality to all of those genes? It really doesn't matter that they're near to each other in the chromosome. but are they similar in function? And this is that paper from a year ago that breaks down the types of functions of these genes. And the one here, the green that are neuronal communication, that's what we predicted when we started this. And I think the important thing about these whole exome studies is they're not based on our preconceptions. They're going to show us what the data show us. The data, we expected this. We expected that how nerve cells communicate with each other would contribute to autism. And you see, it's not even half of them. But I don't want to underestimate that it's some very important ones. Remember I mentioned orexin 1 and SHANK3 had been found with CNVs. This is actually data coming from SNVs. P10 is one that's very important because it's also a cancer gene. So it's one of the genes to find because it has implications for later screening. P10, by the way, is something that is important to test if heads are very large, not just at the cusp that the pediatrician is looking for, but which would be like 2 standard deviations, but these are often 4 standard deviations larger head size. Why are these 102 genes split into 2 groups? Well, the reality is there's no great separation between genes causing ASD and ID. But there was an attempt to say, well, maybe there are some that are found more in ASD than ID samples. We know more about the ID in the ASD samples than the ASD and the developmental samples we know have ASD. So there's a limitation here, but basically showing some genes more affected in one versus the other. You may get an impression that this top purple box has more of these gene expression regulation genes than the ones that occur less predominantly only in the ASD samples. And so this gene expression regulation piece, I really want to emphasize that we didn't go in expecting this. These are genes you expect much more to find in terms of maybe causing cancer, maybe some of them, if you miss both copies, it's probably not survival. But the interesting thing is, and there's a lot of discussion about epigenetics and autism, and these are all basically involved in epigenetics or the control of gene expression. Recognizing these are mostly still functioning, but functioning at half of the dose as would be normal. Probably what's happening is gene expression in neuronal communication is critical that it be fine-tuned and operating correctly, or else the pairing of neuronal communication with experience doesn't work well. Anyway, the point is trying to, at least we've gone from, say, 102 separate genes to saying, well, here's 2 classes of genes. And then we have a few others in here. You see we have 12, 13, we have 11 genes. We're not sure what they do, or at least they don't fit in a nice pattern like the others. So most are related to reduced function, which I mentioned, and therefore most can be studied with knocking down their RNA to produce less protein. And so these become, once we know this, there's a fairly straightforward way to start studying these in cells or in animal models. And so, for example, the first one that was studied of these SNVs, which are implicating only one gene, was CHD8 because it's partly high on the list because it's the most common recurrent one. And basically, people are in labs trying to sort out the specific causes of CHD8 and those other mutations. And so much of the work that was being done to identify the genes is now being shifted to understanding what they do. And frankly, some of them weren't that well studied until they showed up as ASD risk genes. Okay, so a few of these genetic changes are involved in study of how to use that information to treat. Actually, at Rush, Dr. Barry Kravis has been working for a long time and continues to work on moving from gene to mouse to clinical trials. So she's at the clinical trial stage and trying to improve how we test medications that may be useful for Fragile X. SHANK3, insulin growth factor is being used by a colleague at Mount Sinai, has some promising preliminary data on insulin growth factor. It's not a direct substitute for SHANK3, but it's based on understanding how SHANK3 affects neurons. And I'm going to show you a little, just very early work, just to get you an idea both of some glimpses of what we hope in terms of using this information better for treatment. And I'll also mention how it's also a challenge if we have so many different genes contributing to ASD. This is also going to show you a little bit about some of the commonality across genes, even though I focus on this gene SYNGAP1. Dr. Barry Kravis, Elizabeth Barry Kravis at Rush is doing the Fragile X trials. That was one of the questions. Okay. Okay, SYNGAP1 and lovastatin. So I hope you're wanting to go into looking at how a cell's working and how we think that neurons are working to learn, because we're gonna try. So I'm going to show you some slides about this too, but first I'm going to put it in words and try to put it all together. So SYNGAP tonically inhibits RAS when associated with PSD95. So we may come back and forth to this slide. So first of all, PSD95 I also said is MAGUC. So I first of all want to step back a minute and go 10 years ago, really before we had the SNVs, just from the CNVs, we saw this whole chain of proteins that are all interacting with one another. And if any one of them had one of these CNVs, you didn't have to take them all out. You just had to reduce the dose of any one of these, and it would be risk for ASD. So the point is, they're different, but they're all in this very connected, very directly connected pattern. So you see this includes Norexin, which I'd mentioned before, neuroligin, which interacts with it. Actually, the first mouse model of ASD was from neuroligin mutation. When I say mouse model now, I mean someone has a mutation in ASD, and then we take that mutation, in this case, the exact missense mutation, and develop the mouse for this, which has become even easier. And you don't have to use mice. You can use fruit flies and other models. And so this is the presynaptic neuron and the postsynaptic neuron, and this interaction here. The whole idea is more activity across here strengthens the synapse to either continue or to basically be remodeled or disappear. And so any of these missing is enough to cause ASD, and I'm going to focus on this PSD95 or MAGUC and SYNGAP. You see they're interacting here. And I mentioned SHANK, so you see that's part of this, this entire chain. So now we've got PSD95 interacting with SYNGAP. And so what I'm saying is that SYNGAP tonically inhibits RAS. We don't have RAS in this picture, so let's go to this next picture. So this is without a SYNGAP1 loss of function mutation. This green circle here, I hope you can see, it's actually not a circle. It's meant to show that it's interacting with this MAGIC or PSD95. When it's interacting here, it's inhibiting RAS. And so we'll talk about, well, RAS is basically something that's going to stimulate growth of receptors. It's going to stimulate protein synthesis, but it's being inhibited by SYNGAP, by 2 copies of SYNGAP. Now let me go back to previous slide. In this LTP, which is a model of learning, SYNGAP is dispersed from its connection to PSD95 and no longer inhibits RAS. And the dendritic spine enlarges, which means the connection between this postsynaptic terminal and the presynaptic, not visualized here, strengthens. And you actually see that it's a larger spine. It's got more of the NMDA receptors and the AMPA receptors that make any signal from the presynaptic terminal stronger. So typically, this would be an example where that stronger connection would be related to learning some process. It's the best synaptic model we have for how learning takes place. So this is a normal situation where, as I said before, the SYNGAP is no longer connected to this MAGIC, and now only when it's connected is inhibiting RAS. What you see here is some of the SYNGAP that was connected now is floating here, not inhibiting RAS. Now RAS can turn up the protein synthesis and you grow a larger dendritic spine that has more communication of glutamate. Now in this case, what it's showing you is that if you don't have as much SYNGAP, then you have less inhibition of this RAS, and you start out before there's any activity, before there's any experience. Let's say you're learning something from the environment, there's a synapse that should be strengthened, but it's already strengthened without a relationship to experience, and it can't get any bigger. So it is harder to learn, and I will also make the point that you may also have some signals, you may also have some connections that are stronger without experience as well. So that's how the SYNGAP1 mutation leads to problems in learning. And the idea of lovastatin, which is commonly used to lower cholesterol and stabilize plaques. In that case, what you do, you're in this situation, and how do you get back to this situation where you have normal basal transmission? Well, you inhibit RAS directly with the lovastatin, and that's how you try to get this back to there. Okay, hopefully somebody's still with me. So I had a patient. We found this mutation from one of the studies, got it confirmed clinically, and did an open trial of lovastatin in this patient with this loss of function mutation. Initially started at 0 milligrams, went to 20. This is the aberrant behavior checklist. Things improved from 0 to 20. The SRS, about as good a dimensional measure of ASD as we have, not perfect, went down. Improvement in SRS scores, and the restricted repetitive behaviors went down. Went to 40. This ABC is actually the same as it was at baseline, but it involved more irritability than at baseline, so we stopped it because the one thing we don't want is more irritability. And so we stopped it for a while, and then the parents said, let's try it again. And maybe the setting was different. Obviously, things can change because programs improve or have problems or don't, and lots of other factors, obviously, in people's lives. But this time I said, well, wait, maybe last time the problem wasn't going too high. And if the parents would have wanted to try to keep it 20 before, we would have, but they didn't want to do that at the time. So we restarted it, stayed at 20. And in this case, the aberrant behavior checklist came down, hyperactivity, irritability reduced, restriction of repetitive behaviors reduced, and the SRS reduced. I think the most interesting comment from the parents was it was a lot easier to go to Home Depot because although he loved to go there before, he would only want to go and hang out with the lawnmowers. And after that, he was much more interested in more things. Uh, some of the more interest in more things for someone who's about in their 30s is a problem because he became more curious about how keys work in locks. So we almost stopped because he was learning new things. Obviously, you'd want to try before somebody was in their 30s, but that's when we found. Certainly not a huge change in his life, but some improvement. He's continuing now on 10. We're trying to see if that's better than 20. And unfortunately, and this is what I alluded to before, the problem is lovastatin is generic, so we don't have an interested drug company to support a trial. And because SYNGAP1 and most of these are less than 1%, it's hard to get enough people together for a trial. So we're going to have to start thinking about how we do trials more remotely than I think we have in the past, where we would bring everybody into one or a few centers. Okay. This is another example, and this also shows you that there are genes not on the list, but that lead to syndromes. So this person was found in our autism center to have an NR2F1 variant, and NR2F1 means something, but frankly, don't ask me. This is a syndrome that was described as an optic atrophy, where the optic nerve, it's not there and then atrophies like a muscle might, but it just didn't develop in the first place. And it was described as an intellectual disability syndrome, but it came in through a research study. He had graduated from the University of Wisconsin, but not driving in his late 20s, which makes sense because his verbal skills are superior. IQ 141 is nonverbal of 63, and probably makes sense that he takes more Ubers than drives himself. It turns out this is sort of an exception. This gene is very specifically related to the visual spatial pathway, not just the optic nerve, but the occipital cortex as well. And that accounts for this nonverbal learning disability, quite severe nonverbal learning disability. And one of the points of publishing this paper also was to make the point that unless you study more broadly in the population, this had been described as intellectual disability syndrome. He clearly doesn't have intellectual disability. But if you're only, and this happened in the early studies in genetics, if you're only sampling from people with intellectual disability and not the full range, you're going to make erroneous conclusions.
Dr. Cook, can I
Yes,
interrupt you for 1 second?
sure.
We do have a question that popped in the chat. Someone asked, can you talk about CRISPR therapeutic use for SNV?
For SMB,
NV?
for SNVs. Right.
Yes.
Okay, good question. Yeah, let me talk about that in a minute. I want to make a point about how we're going to use this genetic information to develop treatments and where I think that stands in ASD. That's a great question. So if I forget to get to that, remind me. But I think
Okay,
we're about there.
thank you.
So this is another paper. Actually, one of the participants or a couple of participants are involved in writing up this case report, waiting to hear back from the journal. And this came from my clinic, and this is someone was not in our research studies for genetics, but the father connected with the SPARK study. And I should probably put that up later, how to connect with the Spark study, which is a research study, but they do this in a way that if they find something clinically relevant, they do clinical confirmation and get back to people. That's different than people that have active clinical questions where it's better to go to a geneticist and get testing that won't come back in a year or possibly two. But she was identified as having a POGZ de novo loss of function variant, and this is a known syndrome called White-Sutton syndrome. She's in her mid-30s, lives on her own and works with support. She also has bipolar mood disorder and OCD. We're not sure if that's related to this syndrome, but the reality is most of the studies of White-Sutton syndrome have been with younger children and studied less by psychiatrists. So it may be that there's not enough assessment of psychiatric comorbidity, particularly that which develops later. In her mid-30s, she developed worries about constipation, and she developed full incontinence. She developed severe acute prolapse of an elongated colon requiring emergency surgery, fortunately, which went well. She's done well with that. And this particular variant, which is back in that chart I showed you, is one of the top four. Surgical emergencies may occur as often as 15%. So knowing about this means it's a completely different kind of GI concerns and risk than we might have with others. So I think as we know more about these syndromes, we're basically pretty much just finding them now because we're only doing the sequencing to find them. It will lead to, at least for it's probably 5% where we find these kinds of diagnostic variants. It might go up where it may have implications for screening and implications for, what to look for. I would say anyone with a POGZ mutation, again, very uncommon in ASD, but, we identify it. And for those, we might be doing something differently in terms of following up and trying to prevent an acute surgical problem. So, sorry. I think I'll cover the question about CRISPR now. So later on, I'm going to talk about, to me, the point of doing all this has always been to develop better treatments. I would say there are two basic kinds of treatments I think of. The kind we have now are called small molecule treatments, and they're small molecules that can go into the brain. We don't have anything indicated specifically for autism. We have a couple medications that drug companies have gotten to FDA approval for irritability with autism. Frankly, risperidone and aripiprazole treat irritability without autism as well. And so there are a couple of approaches. One approach that I favor greatly, and I have a colleague that's done this with the duplication 15q11, q13 fruit flies and their epilepsy. He's working on this for their autism as well. He's actually developed an autism rating scale for flies called the FlyDOS. But what he will do is he'll overexpress the UBA3A gene, which may be the key player in that 15q11, q13 duplication. And the fruit flies, when they seize, they drop to the bottom because they can't remain in flight when they seize, which makes sense. And so he basically is able to provide, he has them drink a whole set of drugs, seeing if any of them will reduce the seizure frequency. So this is and he's developed some interesting things from that are available drugs that are in a sense possibly repurposed for that specific finding. Now, I think there are two approaches to this high throughput use of what we're finding out from the genetics. And what we would hope is it wouldn't be each genetic syndrome to where we need, we expect to find 200 or 300 of these, not just 100 over the next, probably in the next year or two, as the sample size goes up greatly because of the Spark study. So that's one approach. The one approach is rationally designing small molecules. The number one advantage to small molecules, and as someone who's treated quite a few people, sometimes it works, sometimes it doesn't work, and I like being able to stop something very easily by just not administering. So that leads us to the question of CRISPR and I think gene therapy in general. And the first thing I want to say about ASD is that there's nothing that, CRISPR, you think about by the time, you'd have to do something prenatally, you would have to do something basically probably with fertilized eggs and not even embryos. And one of the points I was trying to make before is we're not very predictive of the outcome of most of these things. Now, if we were talking about Angelman syndrome, almost always Angelman syndrome of the deletion variety is going to lead to a quite severe intellectual disability and epilepsy. But for most ASD genes, we think about the ASD alone, we're not going to be that predictive, even if we test and find something at birth. There's some exceptions, but even with the duplication 15q11, q13 with a high seizure risk, a higher intellectual disability risk, there's a lot of variation we don't understand just from that one finding. So I think CRISPR, which you would really be using, if you're talking about changing developmental course, doing that is probably not going to, I don't see that as quite useful in ASD. I could be wrong. I'm just, it's a question. It's not one right in front of us today, I can say for sure. But I know it's a forward-thinking question. The NIH just had a series of talks about the potential for gene therapy in neurodevelopmental disorders, which would include autism, and they were talking about some of the specific causes, and they were talking about Angelman syndrome. So there are Angelman gene therapy trials for example, Rush is participating. Again, Dr. Barry Kravis is quite an international leader in this way. That's so here's my view of that. You probably have to do it early. And I think that even for most of these syndromes, particularly talking about ASD and not the ID or epilepsy component that often overlaps, it's hard for me to see, unless there are dramatic improvements in gene therapy, that there are many problems of feasibility. Maybe it'll happen, maybe it'll become safer. That may be the only approach that works. In other words, we may not be able to develop small molecules. We may have to develop specific, if these are what's called haploinsufficiency or missing a copy, then increasing expression may be helpful. But here's a very important thing. I was glad Huda Zogby was on that panel, because Huda Zogby was talking about the fact that if you look at the gene MECP2, too little can cause Rett and too much can cause autism. So sure, you want to increase the amount of protein in the right place in the brain. You can probably use gene targeting to do that. Right now, those things almost always have side effect concerns that may improve. Getting it into the brain is a challenge. But the problem is, how do you control that you only restore it to a 2-copy level and not to a 3-copy level? So you could go from a deletion or too little to a too much instead of the Goldilocks approach. So maybe it'll happen. There's a lot of work to be done, but what I'm hoping is people don't get distracted by you find a genetic change, you find a genetic therapy. I think the genetic changes should be used much more to understand how are those proteins causing autism and how do we develop approaches to impact that. So it may be a combination. There are already FDA-approved gene therapies, but they tend to be for syndromes such as a particular muscular dystrophy that leads to death within the first few years. Okay. So these are the current recommendations for genetic testing of individuals with ASD, Fragile X testing, and chromosome microarray, which is just the way to find CNVs. When I showed you that picture with ratios of 16p11.2, that's chromosome microarray. It's been developed more than that picture. However, a recent multidisciplinary consensus statement of geneticists recommends adding whole exome sequencing instead of chromosome microarray, because basically what I showed you is about half of what you find isn't going to be a CNV. And then they were suggesting doing CMA or chromosome microarray only if the exome sequencing is negative. As it turns out, exome sequencing can also find CNVs. It's not as good as a chromosome microarray. In the not too distant future, it'll be complete genome sequencing. It's just a question of cost. And it will capture SNVs and CNVs, and it's a little bit like computers getting cheaper for the same power, and the same thing happens in the genetic sequencing world. Right now, and as it turns out, a lot of the cost of the testing has to do with the interpretation, which is by no means simple. Some ethical implications. We have to acknowledge the complexity of genetic effects. If I made this sound simple, then I failed. The other thing I didn't emphasize enough, but it's an implication of many, many variants contributing to ASD. Well, first of all, I want to make the point that subthreshold ASD traits are often adaptive for individuals without ASD, such as, I'm sure that what I've had to do for research to intensely focus away from people is maybe not even a subthreshold trait. And of course, many of these things may be adaptive to those with ASD. And then finally, if you attempted to eliminate the common variants for ASD, as some people try to suggest might be possible, it's not. You would have to eliminate the species because we all have many ASD variants. And I do think that it has implications. I haven't actually, it makes this concept of pre-existing condition meaningless. We all have risk and are sort of maybe even one straw away from our pre-existing risk becoming whatever the condition may be throughout medicine. And this is, I mentioned this before, so I'll say it again. And this slide comes from a very good question from somebody with ASD at a lecture. And remember, I mentioned these overlapping variants or pools. Let's say 1,000 genes coming together. Well, if they all come together, they might lead to ASD, but by themselves, maybe there's one for language impairment. I don't know what the flip side of that is, but maybe it's nonverbal strength. Anxiety avoids excessive risk, isn't the worst thing in the world. This is the one I talked about before, restricted interest, the ability to focus intensely. That whole set of genes might actually be a set that someone who otherwise might have ADHD can use to function much better. And my favorite is social impairment. This actually came from being in a court with a lawyer prosecutor that wanted to say that the patient who had ASD, that it wasn't relevant. She didn't have ASD. And he basically, what was pointed out to me is that one of the social impairments, and this goes from some tests, task, and just observations, that people with ASD often, it's not that they might not try to lie, but they don't do it as well. Some of that's just good conscience. And that's a quote unquote impairment, but I think there are adaptive components to not lying well. Um, and the other thing is context is essential. So all of these things might add up to ASD that might make it hard for someone to communicate, but the same risk may be beneficial with other gene combinations or even in other environmental contexts. Most importantly, genetic etiology. A lot of people get thrown off by this. They think if I'm talking about genetic risk, then I'm suggesting that something is somehow uh, written in stone, that it can only be treated by gene therapy, but it doesn't at all mean there's a reduced need for rehabilitation, education, non-genetic treatments. I could just add on a whole host of things, recreational opportunities, just it. So the idea is to help empower those with ASD and their families. The common risk variants for ASD are shared partly by all. So, I know parental guilt is difficult to deal with, but genetics should actually be something that reduces parental guilt rather than contributes to it. As I noticed, most of the big effects of genetics are not present in either parent. So to me, the things that may be most useful is identify patients that may have increased risk for medical complications. And I like the question because it leads to this: how do we develop new treatments through the understanding of how these genes lead to autism? It's really our best. I would say it's almost our only angle, and there, I don't want to say 100%, but it's really opened up a whole new avenue of how do we understand how autism occurs, and has at the same time, it's been remarkable. It used to be almost no one in neuroscience was interested in ASD, and now there's a large number of people interested who are doing the very basic work we're going to need to know how the brain works, and particularly to know how changes in those genes lead to risk. So I want to thank a lot of people, all of you who are still on. And a lot of people with autism, their families who participate in the studies, everyone who cares for themselves that they have ASD or others with ASD, everyone who advocates for services and research in ASD, mentors, trainees, other collaborators, several funding sources. And I guess I want to thank my late brother, and I miss him quite a bit. And ready for questions.
Thank you, Dr. Cook. I did encourage everyone to continue to post questions in the Q&A or even in the chat. I also placed the main website for the Spark genetic study in the chat. I hope that's the right one. That is the one by Simmons. So correct me if I'm wrong.
I can't see the chat. Oh, here it is. Chat. Got it. There's a big chat. Here we go.
Yeah, sorry.
No, that's okay. Yeah, that's the one, but put Rush behind it.
Put Rush. Okay.
Yeah, sparkforautism.org/Rush.
Okay. I will do
And
that.
because one of the reasons for the study also, it'll connect people up with larger studies that Spark is trying to match people with. And also Rush, since they're local to our state, may be able to help people if they have challenges working with the larger system.
Excellent. I will do that right now. And I think you've got some questions popping in.
Okay. Is there anything else we missed in the chat? I think that's probably it.
No, Brad asked if the slides will be available, and yes, if anyone is wanting a link to the recording or the slides to review again, please email me or Shannon Dyson, and I will put that in the chat for everyone. And we will get the link to the recording to you so that you can watch again. This was some dense information, so I know I want to re-listen again myself.
Yeah, I want to thank everybody for listening and being interested and for all you do. I'm not seeing questions other than I want to say you're welcome to those that are saying thank you. Are we noticing more differences between presentation of ASD in girls than boys? Need more research in ASD with girls? Yes. Are there any genetic variations we know that may account for this other than the obvious one of gender? So great question. As it turns out, our consortium that's working on, that I show you the 102 genes implicated in the paper from last year. I'm eagerly anticipating a presentation on the analysis of the X chromosome, which I mentioned had been missing. When I showed you the list of, we found things 1 to 22, didn't include X. And so I look forward to that. We do not know why there's a 4-to-1 male to female ratio. It's even higher in ADHD and ASD, and it's even higher uh, the more cognitive strengths there are. I know that there are discussions about whether we're missing some girls with ASD. I certainly know we also miss girls with ADHD and attentive type. And so it's a very active group. Actually, as it turns out, the Simons Foundation had an entire request for applications to try to figure out why there are more males than females. It's not unique to ASD. It's typical of most pediatric onset problems that are not specific to girls. And so hopefully we'll know more about that. It's probably a very good clue. Some of it, I mentioned about 1.7% homozygous X males, but that's not going to count for 4-to-1. So, and just the overall question of ASD in girls is a very good one. What we think is happening is that, we don't know what it is about being female that's protective. One of the interesting research studies by David Skuse in Turner syndrome is fascinating because Turner syndrome is missing an X. And he showed more risk for ASD and Turner syndrome if you were missing the X that came from your father than the X that came from your mother. Now, males don't have the X that came from the father, they'd be females. So that suggests there may be genes that are only expressed on the father's chromosome, similar to the genes that contribute to Angelman syndrome like UBE3A that's only expressed on the chromosome that came from the mother. So that's a potential one, but at this point, it may come down more to proteins and chemicals that are produced in female brains than male brains. There's some hypotheses about testosterone, but it's not so simple, the endocrinology of the developing brain compared to the post-pubertal brain. So that's a long answer saying we don't know, and you're right, we should figure that out. That's like a big question we don't know the answer to.
Dr. Cook, I can't type my questions in the Q&A, but as you answered the last one about the difference in boys and girls, I'm wondering in the Spark study, are there more males enrolled in that as well? So do we just have less female genetic stuff to work with, for lack of a better word. And
Well,
I'm
that's
not a
definitely
doctor.
an impact on that's why with a much larger sample, I mean, the samples that I showed you, we get 102 genes, I forget, but I think the ASD samples maybe 10,000. But Spark has collected fairly quickly another 30,000 or so. So when we go to that much larger scale, not only will we have more samples, find more genes, but we'll have more um, we'll have more females as well. So that'll
Thank you.
definitely help. And there are just some statistical challenges around having only one X for males and two for females. But I have wonderful collaborators, but the one that's working on the X chromosome um, has thought about the complications of that analysis that never occurred to me. But the good news is, it used to be, there were a handful of people working on these problems, and now there's large consortia.
That's great. Thank you. I had one other question about the use of lovastatin with the
I do
syn.
not recommend it other than a research context.
Okay, that was my question, and I'm assuming that you would have to have the genetic testing for the SYNGAP1
Yeah, you'd have
previously.
to know there was a SYNGAP1 mutation. Yeah,
Correct. Okay. Thank you.
yeah. And I mean, I probably, our samples in total, we probably through Simon's and the TASK study and the ACE study, maybe 400 or 500, and one POGZ, one SYNGAP1. That's why we have to really pool across, probably across countries.
Okay.
Uh, oh, good question about environmental components. Clearly, there's work going on in environmental components. I think, and I mentioned this before, that, well, first of all, people are using the finding of their mouse models of valproate exposure and pregnancy based on that risk. Um, and I really think the big development in environmental work is not looking for sort of a common large effect, but looking at it's much easier to do to pool all the genetic variation across the genome than to work on this in environment. But I think people working more to develop what are analogously poly-environmental risk is a major step forward. Um, there are some rare events that cause it um, that are big effects, but it's probably going to be more lots of little effects. And yeah, in some ways, the whole idea, the question of the females is interesting because it may come back to a kind of environmental effect, even though it's genetically driven. As I mentioned before, it may be different hormonal environment and other chemicals. So yeah, there are people working on that. I will be reviewing a paper on that. I'm glad people are looking at that. I mean, the one that is, yeah, it's not a new question, but I think it's been very interesting for people to carefully sort out. And I think it's really interesting. The SSRI risk looks to be related to maternal depression and anxiety rather than SSRI. And that's been an interesting thing that the people studying environment, and I'm glad they're very large and collaborative groups working on environment. I think it's important to do. Theoretically, not all environmental things can be changed, but what you'd hope to find is environmental things that you can change. Um,
Dr. Cook, you also mentioned your study that was in Nature in 2008. And I wondered, you mentioned that you could send that. Would you like folks that are interested to email me, or can they email you directly?
oh, I don't know. I was going to send that one. That's kind of old.
Oh, I'm sorry. Which one were you referring
I
to? The
forgot.
2020 study?
I forgot. I can certainly send you a link to anything.
Okay. If we have questions, I will get folks in touch with you for their specific journal
Yeah, yeah.
needs.
The 2020 study was in Cell. The slide, if you have the slide, you can get to it,
Yeah,
and the other ones you can get to. Not everybody will have access to all of them,
sure.
um, depending on their institutions or subscriptions. There
Sure.
was one that I mentioned and I forgot what it was, so you can let me know later.
Okay, will do. Thank you.
And yeah, thank you all for being interested and what you do. Appreciate it very much.
Thank you. If there's
Bye.
no other
Oh,
questions, I think we'll wrap this up and give you all 10 more minutes of your day. Please complete the survey that will pop up at the end and mark Tap Central Office. We appreciate you so much, Dr. Cook. Thank you for sharing your research and this knowledge with us.
you're welcome. Thanks, everybody.
Have a great day.
Bye bye.
Bye.